It is my great pleasure to introduce today's speaker, a fellow chair, department chair, and a good friend, Dr. Michael Henry. He's professor and inaugural chair of the Department of Biomedical and Translational Sciences here at Macon and John Brock, Virginia Health Sciences, Eastern Virginia Medical School at Old Dominion University. Dr. Henry joined us earlier this year in June 2025 as the founding chair of this new department that bridges the biomedical sciences and translational research enterprises across EVMS and ODU. Dr. Henry brings with him a distinguished record of scientific leadership, academic excellence, and research innovation. Before joining us, he spent more than 2 decades at the University of Iowa, where he was professor of molecular physiology and biophysics and served as deputy director for research and later interim director for the Holden Comprehensive Cancer Center and NCI designated center. And NCI designation of a center is a big thing. There are like 57 such centers, something like that, across US. In these roles, he played a pivotal part in advancing Iowa's cancer research programs, strengthening translational pipelines, and fostering collaborative science across disciplines. Trained as a geneticist and cell biologist, Dr. Henry earned his PhD in biology from MIT and his bachelor's in genetics from University of Georgia. His postdoctoral work and a subsequent research career have focused on the molecular and cellular mechanism of cancer progression and metastasis, with particular emphasis on how cancer cells adapt to hemodynamic forces in the bloodstream, a field in which his lab has been highly influential. His research has been continuously supported by NIH, by Department of Defense, and other national agencies, and his work has contributed to new insights into cancer biology and therapeutic development. Before his academic career, Dr. Henry worked in industry as a senior scientist at Millennium Pharmaceuticals, where his group led preclinical pharmacology and mechanism of action studies of novel therapeutics, including the development of MLN2704, an early anti-PSMA antibody drug conjugate for advanced to phase 2 clinical trials in prostate cancer. Dr. Henry has also been a dedicated mentor and an educator, having trained numerous graduate students, postdoctoral fellows, and junior faculty who now hold leadership positions across academia and industry. He has chaired NIH and Department of Defense study sections and is widely recognized for his collaborative, forward-looking approach to scientific inquiry. Please join me in warmly welcoming Dr. Michael Henry, who will share with us today his insights into translational discovery and innovation at the intersection of basic science, cancer biology, and clinical medicine, with special focus on fluid microenvironment of the circulation. Dr. Henry.
Are we on? Okay, thank you, Rehan, for that wonderful introduction and the invitation to come talk to you today. I really appreciate this opportunity. And as this is a CME event, I have no disclosures. I did not include my slide here, but there are no disclosures relevant to this talk. I thought I'd start out by telling you just a little bit about the Department of Biomedical and Translational Sciences that many of you are familiar with. But this resulted with the merger of 3 prior EVMS basic science departments at the same time that EVMS merged with ODU. So I'm involved in a merger within a merger, which is actually a very exciting place to be because we can really think about how to do things a new way. There are 51 faculty in our department, about equally divided between an education division and a research division. Our faculty provide education across the spectrum of trainees, which I won't rattle off here, but we teach in 8 different programs, including master's and PhD students in the biomedical sciences. And last year provided about 2,500 hours of instruction in 60 courses. There are 4 areas of research focus in the department: cancer, cardiopulmonary and metabolic disease, women and children's health, and neuroscience. And currently, the department has about $16.5 million in extramural research support, with about $4.5 million new just since June 1, 2025. So really proud to be a part of this group and look forward to working together, in part, as Rayan said, to promote translational research. I really think that's an important part of what we do, and hopefully it can involve some of you, students, faculty, otherwise. We're open for business in that area. So I'm going to give a talk, and I'm going to try not to get too into the scientific weeds here. So I'll probably blow through some slides relatively quickly and take any questions about them that you have. There's really 2 simple takeaways I'd like you to get from this lecture. One is that viable cancer cells actively resist mechanical destruction in the circulation. And this was a surprising finding, and I'll tell you why. And the second, which is where some of our work is currently directed, is that having experienced fluid shear stress in the circulation, that can rapidly alter the biology of circulating tumor cells and affect their ability to colonize distant sites. And that shouldn't have been such a surprise, because we know that fluid shear stress affects all sorts of biologies, particularly in endothelial cells, where they're very sensitive to fluid flow and it directs their biology in particular ways. But I'll get into more of that. So I'll start with just a very brief overview of metastasis, of what we think about the process. And of course, metastasis is responsible for about 90% of cancer deaths, either due to the direct effects of metastatic lesions in different tissues or to the indirect complications of that. And this slide just summarizes sort of a classic view of this process, which I'm going to revisit at the end, because there's some interesting clinical dimensions here, which you're a clinical audience and would love to get any thoughts about. So we have a primary tumor here. Cells from that tumor invade locally first into the underlying stromal tissue and gain access to the circulation. My talk today is really going to focus on what happens during this phase when cells are in the circulation, but ultimately they need to extravasate at some distant site, and there they may sit dormant for a long period of time before growing into a clinically observable metastatic colony. And this is an area that we know little about. I would also offer we know little about this because it's very technically challenging to study, which I'll get into. But that's sort of the traditional view of the steps involved in metastasis. So where we come from this is thinking about how this journey through the circulation happens. So this is a bone scan of a prostate cancer patient where you can see lesions in the bone at distant sites. And if you think about the journey that the cancer cells had to take to get there, it's quite remarkable. They would have come up through the venous circulation in the pelvic floor to the right heart, into the lungs, somehow navigating the microcirculation of the lungs, back into the left heart, out into the periphery, probably through another capillary bed. And today it's possible to take a blood sample from a patient's arm and see these very rare circulating tumor cells. So this is a journey about which we have very little information, in part because it's very difficult to study. We do know that these cells are exposed to a variety of challenges, including being detached from the matrix in the solid tissue microenvironment. As I mentioned, they're very rare. Circulating tumor cells are on the order of 1 part per billion relative to blood cells, which makes isolating them very much a challenge. And then they're dispersed into this vastness of the vasculature, about 10 to the 5th kilometers of blood vessels in a human. And so they disperse into that space, which make it challenged to follow the fates of individual cells. So along this journey, cells are exposed to a variety of hemodynamic stresses, which I've summarized here. We've focused primarily on fluid shear stress, which is right here, and that is based on friction between the blood flow and the wall of the vessel. You've all looked at a river flowing, and sometimes the water around here can be challenging, but if you've looked out in a river, you notice it's flowing faster in the center than it is next to the shore, and that's because of friction of the water against the bank. It's the same principle in blood vessels, and the shear is maximal along the edge where the difference in velocities of the streamlines is the greatest, and minimal in the middle. And physiologically, there's quite a range of fluid shear stress that cells are exposed to. It varies over about 4 orders of magnitude, from the highest levels that are the lowest levels, which are in lymphatics and in the microcirculation, to the highest levels, which actually happen in the heart, around heart valves. At every heartbeat, heart valves generate turbulent flows around the edges, which can expose cells to very high levels of shear, but for very short durations. So this slide just imagines what it would be like if you could take a ride on a circulating tumor cell with respect to the forces that they encounter. So as I mentioned, they would enter the circulation and then rapidly go through an escalating level of shear as they approach the right heart, and a rapid de-escalation. This trip takes probably seconds based on blood flow velocities. They would then enter the microvasculature of the lung, where they would become entrapped, simply because cancer cells are generally much larger than the diameter of a capillary. And there they would remain entrapped for some period of time that is very ill-defined. They may die there, they may extravasate into the lung, or they may be displaced to go on the journey now to the left heart, again through an escalating, de-escalating level of shear, and into some other microvasculature. And then, as I mentioned, you could take a sample from an arm vein and get these cells. So while we don't have a precise description of this behavior, at least a quantitative description of this behavior, it's likely to happen this way. But one thing that we are very certain about is that it's an extremely inefficient process. Tumors can shed 10 to the 6th, 10 to the 7th cells per gram of tumor tissue per day into the circulation. And yet we know experimentally and from clinical understanding that only a tiny, tiny fraction of those cells go on to form metastatic colonies that are clinically observable. And so why this is been a big question in the field. And there are a variety of factors that affect the survival of circulating tumor cells, which I've just listed here. The one that we're going to focus on in my talk is cells being exposed to hemodynamic forces. So this project really started out about 15 years ago with a pretty simple question, which is, are circulating tumor cells mechanically fragile? I would say that we and the field assumed that this was the case. And I have a quote from a prominent cancer biologist from a review around this period of time, which basically captured the sentiment that somehow these circulating tumor cells would have to survive these hemodynamic forces. Remember, if your life is in the prostate or any solid tissue, breast, wherever, you don't experience the same sort of forces that you would in the circulation. And because we know that there are features of red blood cells and leukocytes that enable them to withstand hemodynamic forces, it was a reasonable assumption that cancer cells didn't have those and were therefore susceptible to destruction, and that could play into that metastatic inefficiency that I mentioned previously. So we started looking into this, and a former graduate student in the lab, when we looked for evidence for this, we couldn't find any. It's one of those things that made sense at a gut level, but nobody had really ever tested it. And so we started out to do that in a very simple fashion. Okay, I'm not going to try to convince you that a needle and syringe replicates the complexity of the circulatory system. But what it does do is it allows us to apply a known level of fluid shear to a suspension of cells and ask what happens. And we worked with some of our engineering colleagues at Iowa to make sure that we had the math right and could calculate the amount of shear that we're exposing cells to. So this basic model, which will come up again and again, is just using a syringe pump to deliver a constant flow through a needle of a particular diameter. And we can then calculate the level of fluid shear based on the flow rate going through the needle. And so a lot of what I'm going to show you is that this high rate, which generates a range, it's a range, again, because it's maximal next to the needle wall and minimal in the center of the needle. So you produce a range. And this is really extreme. Physiologically speaking, the high range overlaps those turbulent flows that would happen in the heart. And again, for a very brief duration, we call these pulses of fluid shear because essentially you're just pulsing it through the needle. And the amount of time that cells spend in the needle is about a millisecond. So they're very short, brief pulses of shear that physiologically are most reminiscent of those around heart valves. So this was the basic finding that sort of launched this ship. We put red cells and leukocytes from donors into this model, and they actually survived reasonably well. There was some hemolysis that occurred, and that's actually consistent with the amount of force that had been measured necessary to rupture a red cell membrane with millisecond duration exposure. So that actually looked good and serves as kind of a control for this model. At the opposite end of the spectrum were normal primary epithelial cells from mammary gland or prostate. Those cells were remarkably sensitive to this destruction. So after just a couple of pulses, we completely wiped those cells out, fragmented them, and they were gone. What was interesting is that cancer cells from a variety of sources, in this experiment was mostly breast and prostate cancer, but we've since gone on to look at many cells. This is a collection here. And almost all of the cell lines from many, many different tissues exhibit this same level of what we call fluid shear stress resistance. After about 10 pulses, about half of the cells are still surviving, okay? And I want to emphasize that this is adaptation, not selection. And what I mean by that is we didn't just select for a resistant subpopulation that existed within that culture. And part of the evidence for that is if we take the cells that survive this, put them back into culture overnight, and repeat the experiment, we get the same curve. We didn't select for a resistant population out of that collection of cells. So given that we saw that this phenotype was so conserved among cancers that originated from different tissues, we wondered whether it was a property of cellular transformation. And so we looked at this in a couple of ways here. So here we have normal untransformed prostate epithelial cells. And then Bill Hahn, who was at the Dana-Farber, gave us some cells that he had transformed with different oncogenes. So in this case, MYC or RAS, so classical oncogenes. And when you add that to the mix, the cells become more resistant, suggesting that this phenotype is a fundamental property of cellular transformation. Similarly, we did an experiment in melanoma cells where we could conditionally turn on a RAS, a mutant RAS allele, and that conferred fluid shear stress resistance. All right, so everything I've told you so far has been about cell lines. So what about real tumors? So to look at that, we had generated a mouse that has prostate cancer by virtue of having deleted the P10 and P53 tumor suppressor genes. And we took prostates out of these mice, made a single cell suspension, and then performed the same assay that I just showed you. And here too, in cells isolated from primary tumors in these mice, if you have a tumor forming, you have elevated fluid shear stress resistance. Similarly, we had generated patient-derived xenografts from melanoma patients, either from primary or metastatic sites, and we saw the same phenotype there. So two takeaways from this. One, we see this phenotype exhibited in freshly dissociated mouse and human tumors, and it's present in the primary tumor itself. Again, it's not selected for by the process of metastasis. Many of those cell lines that I showed you are isolated from metastatic lesions. So it seems to be this fundamental property of transformed cells. So what's happening? Well, one of the things that we knew could happen is we could damage the cells, and we wondered what was happening there. So we did this experiment with low molecular weight tracers to see if we could determine if we're forming holes in the cell. And indeed we did. And those holes are relatively small. Only the smaller end of these tracers go into the cells. So we're generating holes that are, sub 12 nanometers in diameter. And holes that size can actually be fixed spontaneously in cells just through the biophysical properties of the membrane. We did an additional experiment because we wondered whether fluid shear stress resistance was a property of the cell's ability to repair that damage or resist the damage in the first place. And I'm not going to go through this flow cytometry-based assay that we developed to test this, but the bottom line is that when we looked in breast, prostate, and bladder cells, what was consistent among fluid shear stress among cancer cells relative to non-transformed derivatives of those cells was that they resisted damage in the first place. There was some repair happening, particularly in prostate cancer cells, but we think that it's the ability to resist the damage to begin with that explains this. So with some engineering colleagues at Iowa, we started looking into the physical properties of the cells, and using this micropipette aspiration technique, we were able to pull on the membrane of cells and ask what happens to their stiffness after you expose them to fluid shear. And what this experiment showed is that prostate cancer cells exposed to a couple of pulses of fluid shear rapidly become stiffer. And that was interesting. It suggested that something about the stiffening of those cells was related to their ability to resist damage. And so that immediately suggested some possible suspects in what was going on here. So we know that cells utilize Rho GTPases to both sense mechanical stimulation and then respond to mechanical stimulation. And indeed, that looks like what is going on in these cells. So when you expose them to a couple of pulses of fluid shear, you rapidly activate Rho A in all of these different cell lines here. And I didn't, I'll skip that. So Rho A is at the head of this pathway that affects both the assembly of actin microfilaments as well as actomyosin contractility. So we went about determining whether elements of this pathway were important for fluid shear stress resistance. And indeed they were. And I'm showing you some data here with this agent called blebistatin, which is an ATP competitive inhibitor of myosin 2. And when you treated cells with blebistatin, you reduced their ability to withstand fluid shear stress. So how to test this concept in vivo? So we took a couple of approaches to this, and testing this, the effects of what's going on in the circulation is challenging, and you'll see how we dealt with this. So one experiment is shown here. And in this experiment, we differentially fluorescently labeled a control cell population and then a population that we treated with blebistatin. And we mixed them with these 15-micron fluorescent polystyrene beads, which people use to look at blood flow patterns in tissues because they reliably lodge in the capillary bed. And we can measure the ratio of these cells before we inject them into the mouse. And then we inject them into the tail vein of the mouse, and then as quickly as possible, euthanize the mouse and look at the lodgement of cells within the lungs. Again, when you inject into the tail vein, the lung is the first stop, the first microvasculature that these cells encounter. And then we can look at this ratio of cells again, using the counting beads as sort of a normalizing approach to these in this experiment. So when we did that, and this just shows you what the raw data, we used image analysis to measure the different colors here. And we also included a nuclear dye because we wanted to make sure the cells that we were seeing were intact. And what we see is in cells treated with blebistatin, there was a small, admittedly small, but significant effect on fluid shear stress. That there was an effect at all was kind of amazing because this is measuring the shortest possible trip through the circulation that we can measure. Conversely, we measured destruction of cells using a different assay and found that blebistatin indeed sensitized cells to destruction during this brief trip through the circulation. To take a look at a little longer time frame, we did this experiment where we generated orthotopic prostate tumors in mice that were labeled with green fluorescent protein. And we know that these cells shed a lot of circulating tumor cells as these tumors develop. And then we were able to treat these mice with blebistatin and take a blood draw 3 hours later, and then measure the circulating tumor cells using flow cytometry. And what we found here was a much larger effect. So in the mice that had been treated with blebistatin, there was about a 20-fold reduction in the level of circulating tumor cells in these mice, steady state levels of circulating tumor cells. Interestingly, when we looked at CD45 positive leukocytes, blebistatin didn't really have an effect on these cells. So we think that this, whatever blebistatin is doing here is specific more for the cancer cells and not for the normal leukocytes. So this then was where we were at a while ago. We had outlined this pathway where cells that are exposed to fluid shear stress undergo these mechanoadaptive changes that allow them to survive. And it involves this Rho GTPase, and that is outlined here. But we know that Rho GTPases are involved in a number of other aspects of metastatic colonization in that slide that I showed you at the very beginning. So it suggested the possibility that when Rho GTPase is activated by fluid shear stress, it may be making these cells more aggressive. It may be promoting their survival and making them more aggressive. And so that's where we started pursuing this hypothesis of what doesn't kill you makes you stronger. Indeed, that is what we are finding. So in these experiments, we looked at how exposure to fluid shear stress increased the invasive potential of these cells, which we measure after exposing them to fluid shear and then putting them in an invasion assay. All three of these cell lines are more invasive. And then also, because when you think about circulating tumor cells, they're detached from their normal matrix, which can provide a survival signal and a proliferative signal to those cells. So we looked at how exposure to fluid shear stress made these cells anchorage independent for survival or growth. And again, we found that exposure to fluid shear stress made these cells more able to proliferate within a detached environment, which could be relevant to circulating tumor cells. We then also asked, do these cells actually become more metastatic? And so in this experiment, we used a model where the cells are, this is a prostate and breast cancer cell line, where the cells are already fairly metastatically aggressive. And this is in a mouse with a severely immunocompromised mouse. And we found that, in fact, if you expose the cells to fluid shear stress prior to injecting them into the mice, it makes them more aggressive. Again, not a huge effect, but these cells are already fairly metastatic to begin with. We then used a mouse with an intact immune system, and for various technical reasons, we couldn't look at metastatic colonization, but we looked at their ability to initiate tumors with very small numbers of cells. And there, again, exposure to fluid shear stress increased the ability of these cells to grow into tumors in mice. So what is actually happening here? So the rest of the science here is about trying to figure out what fluid shear stress is doing to the cells that make them more aggressive. So we first looked at the transcriptome, so we exposed cells to fluid shear and then held them in an environment where they could not attach to a dish. It's an anchorage-independent culture environment, and then took time points at 3, 12, and 24 hours. And you can see that a number of genes change over this time course. And one set of genes, this is the genes that are significantly changed according to function at 3 hours. And we noticed a couple of things. One was TNF alpha signaling, so suggestive of some sort of inflammatory response. And indeed, NF-kappa B is activated rapidly in these cells. This is an NF-kappa B activation assay. It's activated even if cells are held in suspension, but more so when they're exposed to fluid shear stress. And of course, that can trigger the release of cytokines and expression of cytokines, which we were able to measure in these two cell lines exposed to fluid shear. Particularly IL-6 is elevated in cells exposed to fluid shear stress. So that was kind of interesting to us because others in the field had been studying the impact of other cells in the blood associating with circulating tumor cells and facilitating metastatic colonization. And what this group had shown is that neutrophils can associate with circulating tumor cells and secrete cytokines that help in metastatic colonization. And one of those is IL-6. And so our data suggests that if cells experience an appropriate shear stress environment, they may be able to secrete this cytokine and facilitate their metastatic colonization alone without the help of other cells. So another aspect of this data is that we noticed that there was a signature that suggested that the cells were triggered to elevate oxidative metabolism. And we measured this in a respirometer and found that actually, in fact, basal respiration in cells exposed to fluid shear stress was elevated. And so we pursued that a bit further and looked at the metabolome. So the way we did this experiment is expose cells to 10 pulses of shear, then immediately plunged them into liquid nitrogen, and then measured the entire metabolome of these cells using LC liquid chromatography mass spec. And so there are a number of metabolites that are changed, as you might imagine, that are represented up here. One of the things that we noticed and expected is that there is a decrease in cellular energy charge. So in cells exposed to fluid shear, AMP goes up and ATP goes down. That makes sense because all of that contractility and reorganizing the actin cytoskeleton is highly energetically costly. So we expected that to be going on here. What Amanda Pope, another graduate student that was in my lab, noticed is that there was, not gonna talk about, wait, where is it? This is the glucose part of this. I'm not gonna talk about this. What I will talk about is in the next slide. What she noticed is that these particular amino acids, serine, glycine, methionine, and threonine, seem to be selectively depleted in cells exposed to the fluid shear stress. And this was interesting because these particular amino acids are involved in the folate cycle, which is depicted over here. These amino acids feed into the folate cycle. And so normally, or at least when I had thought about the folate cycle, I was thinking about generating nucleotides for cell division. Well, this is happening very fast. Cell division is not involved when we're measuring these metabolites. But another thing that the folate cycle produces is NADPH. It can rapidly produce a lot of NADPH, that plays a very important role as an antioxidant defense. And this was interesting because earlier investigators had noted that cells detached from a matrix and in suspension can be driven to cell death by elevated reactive oxygen species. So it's a challenge that cells detached from matrix have. So Amanda went on to look at oxidative reactive oxygen species in these cell lines exposed to fluid shear stress and found that, in fact, those levels of ROS were reduced in these cells. So something about exposure to fluid shear stress is maybe reducing that toxic oxidative burden that the cells have. So as sort of the next experiment, Amanda treated 2 cell lines with methotrexate, which is an inhibitor of dihydrofolate reductase and can block this folate cycle. And what she found, in one cell line, nothing happened, and we still don't understand that. But in another cell line, this breast cancer cell line, methotrexate treatment dramatically sensitized these cells to cell death after exposure to shear. And the experiment isn't in here because it's just been done, but Amanda then went on to treat mice with methotrexate right at the time that we introduced the cancer cells to them, a single dose of methotrexate that's non-toxic. I should emphasize that. The dose of methotrexate that we're using is non-toxic to the cells alone, but if you treat them just at the time that you introduce them into the mice, it sensitizes and it reduces the metastatic burden that these mice face. So this slide just summarizes some of these changes in metabolism that occur very rapidly after exposure to fluid shear, which we think ultimately contribute to the ability of these cells to productively contribute to metastatic colonization. All right, so that's was a quick run through of some of the science here. And I just want to spend just a few minutes talking about what are some of the clinical implications of this research, because what we would like to think about doing is finding a way to turn this trip through the circulation into a more even more of a challenge for cancer cells, potentially by disabling their ability to resist fluid shear stress. Methotrexate might not be the best drug for that. It's an anti-cancer drug in its own right in some types of tumors. It's also used in inflammation. There are side effects associated with it. But if we can understand these mechanisms by which cells survive this trip through the circulation and disable it somehow, we might make that trip through the circulation even more of a barrier to metastatic colonization than it is, make it more inefficient. So that's at least the concept. But there's a lot of resistance to thinking about intervening in any aspect of metastatic dissemination. We're only talking about one of them. You could think about blocking invasion, any of those steps related to metastatic dissemination. And there is, quite frankly, we have really never explored those sorts of therapies. And there are reasons for this. So, but I'll just say at the outset, we have been progress in durable treatment of metastatic cancer has been very slow. There are successes, certainly with immunotherapies. For some patients, they can yield durable responses. Targeted therapies can have a period of beneficial effect before resistance sets in. But still, we need to come up with more, with better treatments for metastatic cancer. And so it's the case that most therapies are typically evaluated for their ability to shrink pre-existing tumors. So we don't even typically think about blocking any of these steps before there's a clinically observable metastasis. And part of that is that there is this sort of long-standing idea that dissemination has already happened. The patients have disseminated disease that you can't see clinically. And so that falls into the bucket of the horses out of the barn. It's too late to do anything about blocking the steps in metastasis. So you really need to focus on the disease that is there. Also, there are technical issues coming up with clinical trials to evaluate something that might prevent metastasis could be very long and expensive, and that is certainly a challenge. But there are some clinical scenarios where you could think about the value in trying to intervene in metastatic dissemination, and I've just listed some of them here. So treatment of non-surgically manageable primary tumors or solitary metastatic lesions. Treatment of patients at high risk for invasive malignancy, treatment immediately following surgery, where it's been clearly shown that the surgical manipulation of the tumor can release a shower of circulating tumor cells. It's not so clear that that contributes importantly to metastasis, but at least it's a point in time where you know that there's an abundance of circulating tumor cells. This one is interesting. Treatments of patients with hepatocellular carcinoma following liver transplantation. So one of the complications there is those patients often have disseminated disease elsewhere in the body that will then recolonize the transplanted liver. So there you have a point in time, you do the transplant, you know that for that complication to exist, cells have to get back into the transplanted liver. So that would be a point in time that you could evaluate that. And then I think the final and perhaps most profound argument is still really conceptual at this moment. Using something in the adjuvant setting to inhibit what is, I'm calling, and I'll show you, is tumor self-seeding. This is a very different view of metastasis, and it's summarized in this slide. So I showed you what the classical view of metastasis is, with these steps that sort of happen one after another, leading to a metastatic lesion. This is typically thought of as a unidirectional process with a fixed start and end of dissemination. It goes in lockstep from here to here. So a surgeon, Larry Norton, and Joan Massagey, who's a cancer biologist at Sloan Kettering, developed this concept based on experimental data a while back called metastatic self-seeding. And in this scenario, there is a continuous release and trafficking of tumor cells from the primary tumor to metastatic sites. These cells can then traffic back to the primary tumor. They can traffic between metastatic lesions. And this continual trafficking may drive some of the evolution of the tumor in these sites. So even if you have an established metastatic tumor, there may be an ongoing issue related to this trafficking that could affect, that if you could block that trafficking through the circulation, you could have an effect on metastatic outcomes. So if this model is true, you could potentially intervene in dissemination even much later than when you have identified the patients that have at least a limited number of metastatic lesions, which might be operable, but you tend not to do that because the assumption is that there's disease elsewhere and the risk of the surgery isn't worth the disease popping up in lots of other sites. So I think this is still a provocative idea, but it could lend support to coming up with some strategy to intervene in metastatic dissemination, even when there may already have been some initial seeding that takes place. This phenomenon too can explain potentially why it's been so difficult to understand the genetics of metastasis. It's possible that these cells are moving around, and metastasis can be oligoclonal. And it's not that the cells have to arrive at the same site at the same time. They may come in at different times, populate the metastasis differently over time, and lead to a very different genetic outcome than thinking of a single cell that makes it through this and establishes a metastatic colony. So just one of the other implications of this. So I think I will stop there. I'd like to thank a number of collaborators at Iowa primarily, but also some other institutions, folks in my lab that have contributed to this. And actually, this slide needs to be updated. The only current person in my lab is Dr. Dykstra here, who came from Iowa. And we've had funding from various sources for this project over time. So I would like to thank you for your attention, and there's a little time for questions if you have any. So thanks.
Go ahead, please.
Thank you. That was completely different. If you want to reduce stress, which makes potential cancer stronger, there is a bit of research about using beta blockade in breast cancers, which tends to improve survival.
Yes.
Do you feel that this is relevant to your research?
I do think it's relevant. And in fact, one of my collaborators, Amy Rowat at UCLA, she studies beta-adrenergic signaling in cancer. And in fact, they have found that can contribute to the stiffening of cancer cells as well, and that beta blockers may disrupt that. And we've collaborated with them to look at the specific phenotype. And so I do think that there might be something there like that. That could be an example of something that might be an intervention worth trying, or that one could simply look into the medical literature or the medical record and see if there's a relationship between development of metastasis and patients that are on beta blockers. We've thought the same for methotrexate, mainly for patients with being treated for inflammatory conditions like arthritis. Can you look at something in the record there that would give you a clue that exposure to that sort of agent could have some beneficial effect in metastatic outcomes? So yeah, a beta blocker would be much closer to some sort of drug that you might want to to give in this indication. Yeah. Yes.
Dr. Arabi?
Thanks. Very interesting perspective. And to follow up on Dr. Flanner's question, does hypertension increase metastatic disease?
It's a good question, Paul. We don't know. There is some work that has been published that suggests that elevated exercise, so increasing hemodynamic stress through that means may have an effect on the ability of cells to survive. This is using a very different model than we use to do this. But simply looking at the relationship between hypertension, what I don't know is how much. I don't know how hypertensive states with the the effect that has on hemodynamics, how much that increases the level of shear that these cells might experience. It's something to kind of maybe look into first, but it's a good idea. I mean, anything that might influence that flow rate, essentially the stress that they're feeling, in principle, could affect things. Now, you might think that beta blockers would be working counter to that because they may lower blood pressure. But yeah, we don't know. Yeah.
Dr. Carbone?
You had a couple of questions, and apologies, I missed the first few minutes, so hopefully that was not covered. But for the first one is, I come from more cardiovascular research.
Yeah.
And so I remember years ago, there was a thought that laminal stress versus more of a oscillatory pattern that would cause a different pro-inflammatory phenotype, essentially. Do you see the same, and is there a dose dependent with the oscillatory and then maybe IL-1 beta or IL-6?
Yeah, that's a really good question, and we haven't really explored that yet. So I'm familiar with that literature in endothelial cells, like how they experience laminar versus turbulent, flows, that this can affect the inflammatory processes related to this. We haven't yet looked at that. One of the things we're actively doing now is seeing if we can develop a model where and we're having to change the way the needle and everything is constructed, where we can expose cells to lower levels of shear and where we can vary the exposure dynamics a little bit along the lines of what you're doing and explore how that may influence the biology that we're seeing. Everything I've said here is really pushing things to an extreme, which is what you missed at the beginning when I came in. We came into this thinking believing that cancer cells were going to be susceptible to this. And what we found is that they weren't as susceptible as we thought. And so we really wanted to push the envelope with this extreme amount of shear. But I think getting into more physiologic realms, and unfortunately I wish there was, but there is not a good in vitro system where we can really explore the entire, spatiotemporal range of cardiovascular dynamics. It would be great to not have to do something in an animal, which we eventually, have to do to test these hypotheses, but have a more sophisticated in vitro system where we can vary the rates, the durations of pulses, the levels of shear and things like that. And we don't have anything like that, but it's a good question. And so, yeah.
Other questions?
Yeah,
go ahead.
I had another one specific to inflammation. I remember years ago in cardiovascular disease, there was a study called the CANTOS trial where they used interleukin-1 beta and showing reduction in cardiovascular events. And one of the sub-analysis showed some reduction in lung cancer-related outcomes.
Mm-hmm.
Then they did a lung cancer-specific study, metastatic lung cancer, and they didn't see a benefit of, I think they used canakinumab, IL-1 beta monoclonal antibody.
Mm-hmm.
One of the concern there said, well, probably the disease was just too advanced and too severe to intervene with an anti-inflammatory strategy.
Yeah.
And so do you see a point in which it's just too late
Yes.
to use those, or is there a point in which through the disease course where we should intervene with some of the anti-inflammatory?
Yeah, I think was layered into the comments that I made at the end. Some of these processes may be very important in the early hours, days, weeks, when cells initially start to form a metastatic colony, when their survival is probably the most tenuous, bringing in some of the biology that inflammatory mediators could help promote, that kind of metastatic niche that develops. That's, to me, when it probably makes the most sense. But as I said, a lot of anti-cancer strategies are tested for their ability to reduce observable, imageable tumors. And so some of this may be beyond, it's just exactly as you suggest. It may be too late to intervene in the biology there that really matters. And so you have to think about doing it earlier and measuring it in some way that looks for prevention of metastatic lesions rather than shrinkage of existing metastatic lesions.
Yeah, one comment, and that is, all this seems like started from trying to find evidence for sort of thought of an accepted wisdom.
Yes, yes.
And when you started looking for evidence, you actually found evidence not supporting that the established, now we can call perhaps dogma. So for students and others, I think that's a good point that sometimes we do think about things without any evidence as established truth and fact. But that's just one example where it didn't work out that way.
Exactly, and that launched this whole ship. I mean, in fact, the student was very near finishing his thesis, and he thought he was going to have a quick paper showing that what everybody thought worked that way, and it turned out not to be that way. So yeah, we got going down this path. And so.
I see Dr. Casabo has a question.
Thank you. It was very thought-provoking talk. So the teaching, at least in my medical school, was the main way that cancer spreads is through the lymphatic system. So it's very interesting to see how it goes through the systemic circulation. As far as you know, is there similar or parallel research going on to look into how it moves through the lymphatic system?
Right.
And if that shear stress is similar?
Yeah, so it's a really great question. So to get to really distant places, you have to go through the circulation. There's no lymphatic, route that you could follow, like in that patient with bone mets, from the prostate to those very distant lesions. But you're right. And this is happening, and that's why we go get sentinel lymph node biopsies to know whether it's happened. It's a place where you can go look for metastatic dissemination. I would say there is evidence that going to lymph nodes is not an obligate step in distant metastasis. There is evidence that you can spread directly into the circulation, but going to lymph nodes is probably important in a variety of ways. It can affect how the immune response is going. There's even evidence that cancer cells that go to lymph nodes first can rewire their metabolism, again, around this folate pathway that enable them to withstand their subsequent challenge through the circulation. So the flow dimensions are incredibly different. It's much lower levels that go through there. The levels of flow in lymphatics are so low that they would not be damaging cancer cells in the way that I showed here at this much, much higher level. But flow is probably doing important things in lymphatics, like it's affecting how tumor antigens may flow into the lymph nodes and affect things. But we have focused sort of at the other end of the spectrum, thinking about what high levels of shear would be doing. So, yeah.
Thank you. This was a great talk. Thank you very much, Dr. Henry.
Yeah. Thank you all.